For this video, we're going to talk about translation. This is the latter half of Grandma's cookbook. This is about what happens when you get into the kitchen. So when you get into the kitchen, you start here, and you have two ribosomal subunits. Now these two units will come together and attach to one end of the mRNA. So they're going to attach, and then the tRNAs, like the children, are going to bring the amino acids to the ribosome. And this is what we're showing in this video. As you can see, the amino acids are being accumulated here in order, and basically the transfer RNAs, these are the T's here that I have, and they're a different color, so realize each one is a different type of tRNA, shaped a little different, structured a little different. And what each of those are going to do, they're going to attach to a different type of amino acid. Now there are 20 amino acids that you can use to put together a protein, just like there are 4 bases that you can use to put together to make DNA. Here we have 20 particular amino acids that we can use, and I've represented them here by color. We're only going to do a couple of them here, but the idea is the transfer RNAs attached to particular amino acids. And so here we have a transfer RNA dropping off this red amino acid. All right. And then once all of this is done, everything just kind of breaks apart and goes its own way. The protein can go do what it needs to do. The mRNA may be destroyed, or it may continue. Another ribosome may attach. So here we have the two subunits of the ribosome coming together on one end, and it's going to read the letters 3 at a time. This is called a codon. That'll be important for some worksheets that we do in the future. But you need to know that it reads them 3, and then it's going to jump to the next 3. And whenever it jumps to each 3, a tRNA, these little T's, can come in and bring an amino acid to the ribosome. And the ribosome will stick that amino acid onto the end of this growing chain. So here we go. Let's start from the beginning. You have 2 ribosomal subunits. Here's our messenger RNA, and basically they're going to go attach to one end of it. And so the 2 subunits come together to form a functioning ribosome. Now the functioning ribosome then waits for a tRNA, and it will, according to these 3 letters, that tRNA is going to attach. So here, different 3 letters, so it's a different tRNA, and that tRNA dropped off a different amino acid. Now we have a third set of letters, so a different tRNA, and it dropped off a different amino acid. And finally, we have our last 3 here that drops off this red amino acid. And then we're at the end, so everything breaks apart and everything goes its own way. So let's go through this real quickly together as one giant loop. We come together. First thing, 2 subunits form on the end of the mRNA out in the cytoplasm, and they attach at the end and read the first 3 letters. The tRNA drops off the amino acid. Another tRNA that is associated with these 3 letters drops off a different amino acid. And here we have a different 3 letters, so a different tRNA is going to drop off a different amino acid from the first 2. And then finally the 4th one, and then we're out of letters, so everything is going to break up. Now I've simplified everything here. There are a lot of things that go on at the beginning, all right, and technically this particular mRNA would not work. That's because the first place that it's going to start putting together amino acids is at an AUG, and this is an AUC. So therefore, technically, this mRNA wouldn't work at all. It would be non-functional. And we'll talk about these things later. For this video, all you need to know is this general process. You're sitting here looking at this mRNA, and you need to understand that out in the cytoplasm, the ribosome is going to attach to one end, all right? And the tRNAs are going to bring and drop off the amino acids to the ribosome, and the ribosome is going to stick them together to make our protein. Now technically this is not a protein yet, it's just a bunch of amino acids stuck together. To actually get a functional protein, these amino acids, here the pink one followed by, let's say I think it's blue, right, and then I think there's a yellow and then a red. Well, they have to fold up. They have to fold up into the right shape. and the shape is very important for this protein to function correctly. So here we're just assembling it. We're putting the parts together, but you have to mix it up correctly. And that is also very important. Just realize that this is not a completely accurate depiction. The whole idea is just to get an idea of the parts and then how they come together and work. That you kind of need animation for this. So that's why I did this one, trying not to give you all the details all at once. There are several more details. One thing you should be aware of is that the ribosome does read the mRNA 3 letters at a time. So that's one of the main things you need to get out of this is that the ribosome attaches to one end, all right, and then it kind of pauses at every 3 letters, and it waits for a tRNA to come and attach and drop off the amino acid. And once it drops off that amino acid, then the ribosome will attach it to the growing chain of amino acids as it moves along. So this kind of reminds you of how RNA polymerase works. So just like RNA polymerase reads the DNA and makes this copy right here, the ribosome attaches and it reads the mRNA, 3 letters at a time though, and for each 3 letters it attaches, a particular tRNA will attach and drop off an amino acid in which the ribosome will attach to the growing chain. Here's our final chain. This will go off and fold up to make a functional protein.